Well, thank you very much. First, I should start with the acknowledgement to the Oceans and the OCEANS'16 Organization Committee to invite me to give this keynote speech. Well, I'm an oceanographer, so I do science. So actually, I feel really honored to be here. I finally have the chance to tell engineers how wonderful I feel when the instrument works, how frustrated when the instrument does not work. So especially in the rough sea and the pitching rolling and you're seasick, but meanwhile you have to fix your instruments when it's dead. So, but before I start, I would like to have a couple minutes to give you an introduction about the Institute of Oceanology at the Shanghai Jiao University. The institute was established 3 years ago with a mission which is exploring, understanding, and predicting physical, chemical, and biological phenomena and processes in the sea. And serve the resource exploration, also develop the technologies engineering for the ocean observations, sampling, and explorations. So in these 3 years, the institute started from 4 people and grew up into 26 this year. And we had about 90,000, I should say 90 million research grant from the Chinese government. So we are doing very well, and we are doing the science and do the engineering and do the technology. And hopefully, we will be a home for the innovation and creative thinking. So when you have a chance, when you come to Shanghai, stop by Shanghai Jiao University, visit the Institute of Oceanology. So now I want to start with my talk, which is advancing integrated environmental sensors for the multi-scale ecosystem. So we're facing global challenges. And the first is that environmental is degrading, and because with the population increase, industrialization, and we put the pollutes into the ocean, and we take the land from, we take the coastal area from the ocean, and the environmental is degrading. And we also facing the resource depletion because we all know that overfishing really destroyed our coastal ecosystem. And also we're facing climate change. We know global is warming and it become a fact. So we need all the sensors to measure the state variables. We need sensors also measure process rates and for the exploration. So I want to cite this nothing begets good science as much as development of a good instrument by Humphrey David. So because we really need your help in the science to measure those things we really desperately need. So in the last few years, we are fascinated by 2 things. One is that we hope we can go into the ecosystem-based management. We need the models from physics, biochemistry, all the way to the fish. And then we have the global warming, and with all these models from IPCC to predict the ocean will be, or the atmosphere will warm up, the Earth will facing a deficit in the future. But if we look at them more carefully into all these models, especially in the oceans, we start from the circulation models and to biochemical models, and then we jump into fishery models, and there's nothing between. So it seems like you can predict the fish population, predict their biomass without really link to the primary production, link to the physical processes. So what I want to say is that a predicting model or prediction of the nature should be meaningful only if an instrument can be made to observe the nature first. So this I really want to tell you, I need, as a scientist, really need the help from engineers. So I'm going to give my talk based on the project funded by Chinese Ministry of Science and Technology as a 973 plan project. It is the AMBO endorsed project now. The title is Living Resource and Ecosystem Dynamics on the Slope of the South China Sea. So the major focus of our study is the middle pelagic fish, and we know it's getting more popular these days in Europe and also in the United States. So we're looking into two things. One is the resource, and the second is the climate, the carbon sink. If we look at the global of the biomass estimated by combine all the data at the United Nations, which is about 1 billion ton as a wet weight. So think about global fishery, which is 0.08 billion ton. So 10% of the middle pelagic fish biomass, which really can support enough fisheries we demand. And we know about 95% of the middle pelagic fish never touched in the global ocean. Actually, probably 99% never be touched. So, and also we have to notice, and in the recent estimate, the biomass of middle pelagic fish is probably 10 to 100 times underestimated. So if we took a real number, so the number could be a lot higher. So in the southern China Sea, the historic estimate, which is about 30 million ton wet weight. If we look at the Chinese fishery, which is about 2 million tons. So therefore, 10% of the middle pelagic fish in the South China Sea can really meet the demand of Chinese fisheries. But of course, there are different fish. Maybe not the fish we would like to eat, but it's a potentially strategic biological resource. So with Chinese estimate, the fish demand in 2030, the Chinese population will be 1.6 billion. And the annual fishery addition we need is 2 million tons. So with the biomass we have in the South China Sea, actually we can meet the demand. So the two figures I showed over here, so basically it's the older data set and from United Nations, and this is the Chinese surveys. So we can see it clearly, so that in the China Sea, the measurement over here, which is about 2 or 3 times higher than the historical data. So the project is looking into it, would it really support basically this middle pelagic fish in the South China Sea, and try to think about what we really mean the resource, how the ecosystem can support this fisheries, and also what's the climate impact of this by this middle pelagic fish. So this is basically our scientific questions, basically looking into the ecosystem and also its effect on the carbon flux. But if we look at this slope ecosystem, which is very complex, and we start have this, the focus is the middle pelagic fish, but we should start from primary production. Do we have enough primary production actually support these fish populations? And we know the middle pelagic fish, they do not eat phytoplankton. They have to go through the zooplankton. And also the middle pelagic fish, they move up and down every day at the surface at night and feed, and then go to deep water about 600 to 800 meters, and they rest during the daytime. So, and we hope we can make all the measurements to understand the ecosystem, look at the contribution of middle pelagic fish to the biomass and also the carbon flux. But if we think about this, we need to measure all this from physical processes to the nutrients, to the phytoplankton food web, and to the fish food web, and then into behavior of their migration. And finally, we can end up in some models to predict what's going on. So, which is a really complex system. So in that way, one sensor won't help. We need an integrated sensor system. So we start to design this project, start from basically numerical modeling, look at the circulation patterns in the South China Sea slope, and look at the trajectory of the water mass through some drifters and with the modeling. And we use the water sampler, take a water sample, so meanwhile take all these hydrographic measurements and all these nutrient measurements. We do the nettos for the planktons, and also we do chore for the middle pelagic fish. And we have the lab experiment and also lab analysis for all those parameters we need to study. So we made a list of the instruments we need to measure them, from physics to the chemistry to the biology, which is, this is actually, it's a summarized list. If we expand the list, we need about 3 or 4 pages of these parameters. So the challenge is quite big to do all this, and on the ship at the same time, and hopefully at the same place. So what we did is we built an instrument package with all the sensors that we can think about this to those variables we need. We have the physics, which is the CDD, of course, very typical. We have the lowered ADCP to measure currents. We have the path sensor measure lights, and we have the transsometry to measure the transmittance of the water. The chemistry, we measure the dissolved organic matters and the dissolved oxygens, and color the dissolved organic matter. For the biology, we need to measure fluorescence, and we need to measure particles in different size, and from bacteria all the way to zooplanktons. And then we take the water samples, we analyze nutrients, DOC, POC, and all kinds of variables. And because those are very important to the biogeochemical cycling. And the biology, we have microbials and phytoplankton and macrozooplankton and mesozooplankton, all things. So we hope we can everything kind of combine, build onto this CTD rosette. So in that way, we can take all this sample measurement at the same place, same time, without have to mismatch them. So we did all this integration onto one, which probably the most expensive CDD ever built. And we built a lot of CDDs in the United States, in France, but this is really the most complete, the package. So this is a couple of photos to show you how we did it. And we're on the ship, and it's the fish trawler. And we take the CDD sampling, and every time we have a team of young students, enthusiastic students, come to take all the water samples, work 24 hours a day. And then we take nettles, especially we take nettles at night, because we know you never catch a fish or catch a zooplankton during the daytime. And we do trawlings and different depths. So, and one thing I have to say is we really caught a lot of mesopelagic fish, but the people ask me why we didn't catch them in the past. So the answer is instrumentations. We had a fantastic bioacoustic on the ship, which really tell us where those mesopelagic fish. So these are all the mesopelagic fish. They stay in the deep water in the daytime, and they move to the surface. So when we tow our net, we go through these layers. We do not tow our net, go through the empty waters. This is why we tell the fishery people, say, the reason you cannot catch them, because you do know where you catch them. So now we have acoustic, we can see where they are. We can catch them exactly. This is how wonderful it is for the instrumentation. And use a model. Model is also we can think about, which is the technology, which is very powerful. And then we use this called Lagrangian models to look. We use the satellite altimetry data to look at the surface circulation, and then we put the Lagrangian particles into it. We look at we call the longitude advection and the latitude advection. So we can look at the water source, where did it come from? Does it come from the coral shield? And also we can look at its latitude advection, it come from the shelf area. Look at how this water mixed in our survey area, so we know exactly where they are. So, and when we talk about the technology, and I think modeling deserves a space, which is also a very, very powerful technology to tell us the circulations. So the fish acoustics really gave us a fantastic estimate. So during our surveys, our low estimate, which is about, during first cruise, which is 35 grams per meter square, and which is about 10 times higher than historic estimate. And in the second cruise, we had 28 grams per meter square, which is basically about 7 to 8 times of the historical measurements. So which is really, really high. So the biomass in the South China Sea, which give you about 100 to 120 million tons. And I have to say, this estimate is called the lowest estimate, because we know the acoustic models, they give you a very large variation. So we collect all these variable acoustic models, we run through it, we pick up the lowest estimate. So this is what I say, this number, which is really at the low end of estimates. The real estimate, we do not know where it is, but it could be a lot higher than the number I give over here. So we have really a good estimate of the biomass of this mesopelagic fish. Without acoustic, we really cannot solve the problem. This is why I really, really feel excited about the achievement of acoustic. The second thing is that we look at the carbon export into deep ocean. So we know the Southern Ocean is with the highest carbon export in the world oceans. And if we look at the historical measurements, So in the Southern Ocean, we say average is about like 15 over here, 15 millimoles per meter square per day. It's right over here. But this export is only down to 100 meters. In the 100 to 200 meters, most of the particles, the POC, will be scavenged by whatever the biochemical process, which is down to 200 meters. The number is a lot lower. But we estimate our export by this middle playa fish is about 13 millimoles carbon per meter square, which is down to 400 meters, which is a lot deeper than that. So the middle pelagic fish contribute to the global ocean could be very significant because we know you can find the middle pelagic fish almost everywhere in the ocean. So people won't be able to see this carbon exported by the middle pelagic fish until we had our list, which is the laser in situ scattering transitometry. We had really measured so we have the middle pelagic fish, the rest during the daytime at this depth. We can see here, you can see the POC get a maximum lump over here, which is really the first time people ever see how this POC related to the middle pelagic fish. So this is why instrumentation is really one of the important factors can help us to really see what's going on in the water column. And we take a lot of measurement from the nutrients to primary production and from primary production to the secondary production. And we have this, the primary production incubation experiments. We have the nutrient measurements, and also we have the trace metal ion measurement. And we did a lot of this incubation experiment. So what I see is I really cannot see any correlations between them. It is so chaotic. So now what we can do about these chaotic measurements because they are not really simply correlated. So this is why we need people to help us to solve this because we never be able to measure them at the same time because we do not have an instrument that we can really measure the primary production in the water column. So we basically try to also look at the tropical processes, how the fish and the middle plankton and the primary producers, how they correlated. So we had to estimate it's about the phytoplankton, which is a 2 gram carbon per meter square, and the zooplankton is about 1 gram, and middle pelagic fish is about 3 gram carbon per meter square. But if we look at the vertical processes, the primary production we measure is about 400 milligram carbon per meter square per day. So fish, to maintain this population, it need about 0.15 or 150 milligram carbon per meter square per day. So which is about 30% of the primary production. But we know you go through primary producer to the second production, which is zooplankton, which generally people say it's about 10%. And then from zooplankton to the fish, another 10%. So basically the biomass transfer efficiency, people usually think it's about only 1% from the primary producer to the fish. But now we need 30% of the primary production to the fish. So where the energy come from and how the biomass can be balanced and how zooplankton play the role in this biomass transfer and make the ecosystem more efficient. So that is something we do not know. And we don't have any method actually these days can really sort this out. This is why we say that we need really the help from engineers, can build some fantastic sensor, can help us to sort this out. So I do a primary result summaries. So we studied the middle pelagic system, and the surface of the Southern Ocean is really desert. And if you have been on the sea, I can tell you, I have been on the sea, it's total different feelings. When you're on the sea, you always see seabirds flying on your, on the back of the ship and going back and forth. But if you go to South China Sea, you do not see a single bird. It is a totally desert. If you measure the fluorescence, it's down to 0 on the surface water. It's so transparent, and the optical depth is super deep. But underneath of it, there is a very active, very large middle pelagic ecosystem. And the fish resource, we estimate, is really high, which is about 28 to 35 gram carbon per meter square, which is a really, really high biomass. I'm sorry, it's not carbon, it's a wet weight carbon. So, and the carbon export also to the deep water is really, really high. But there is one question we cannot answer, which is the fish require 35% of the primary production. But the fish cannot eat the primary producer. But where the energy comes from, and we have no way to sort out in these days. This is why we have missing parts in our measurements. The missing parts, which is the first, is that we have no time to repeat our measurements. So if you ask me, what is your standard deviation of your measurement, I say, I don't have it. You can ask me the accuracy or the precision of your instruments, but I can tell you the nature variabilities, because only once you measure, that's it. It takes our 20 days to get the survey done. So we needed some fast those sensors that can measure a lot of time in a very short period. So we cannot measure the trace matter. We cannot repeat the measurements of trace matter, nutrients, phytoplankton, zooplankton, and abundance. But worse is we do not have any way to measure the process rate, so which is the transformation of different materials. So we do not know how to measure nutrient removal rates and the cycling rates and the biomass transfer efficiency between trophical levels. And we have no idea what is the food web structure. So we're advancing all the sensors and technology, but we have some fundamental things we really cannot do and cannot resolve them. Say, for example, all these surveys we only can be done at once. Seems like the primary production is very well with this ocean color, but when we look at the nutrients and the ions, they are totally in stochastic patterns. They have nothing really coherent. And all these measurement of phytoplankton and also measurement of this primary production, and they're only ones. We do not know how they vary with the time, and there's no way we can repeat them. So we need an environmental sensing system, which is we need to put everything onto the system, which is we call the state variables. Here I made a simple list, which is we start from the CDD rosette. We have the temperature, salinity, currents, so we can do it today. And then we can need some optical properties, which we can put the AC9 and all this onto it, all the power sensor onto it. And then we need the nutrients, and we need the nitrogen, phosphate, silicon, and the PCO2 trace metals. I know people have been working really hard on those macronutrients, but all this still distance away. And we need to measure organic matters, which is the POC, DOC, because those are really important. It's a fundamental for microbial loops. And without them, we cannot tell you the food web structure. And we needed the biology sensors, which can measure phytoplankton, zooplankton, and nectarines, which can tell you at least what's in the water, what's the ecosystem structure. And we needed to measure the abundance. And these days, the DNA, RNA sensors, the microchips are getting very popular. How can we put it onto this package? And of course, we can list the geologies, which is the sediment size, types, and the density. And also, most important, the settling velocities. Now imagine we can have sensors these days put onto the CDD rosette. It's a matter of you make the rosette bigger. But also you can imagine put onto a moorings, put onto a container ship. But we will have challenging to put them onto a towed vehicle, and more difficult to put it onto this unmanned gliders or AUVs. And if I remember the talk just a few minutes ago, which is the payloads of a glider, which is made of a few kilos. But if you want to add all these sensors onto one of these platforms, it will be really, really challenging. But without this, we cannot tell you what is the current status of the ocean, the ecosystem. But more importantly, we really do not have very many sensors actually measure process rates. So we need a removal and transform rates of nutrients and trace metals. We need a transform and the transformation rates of organic matter through biochemical processes and between trophical levels. We need a flux measurement between air, sea, and the sea sediments. And we need an instrument to measure behavior of plankton, fish, and mammals, how they respond to environmental variables and the predator and also life histories. And we need to revolutionize our sampling method. Say, for example, in these days, the nettle still is one of the primary sampling methods for the zooplankton and the fish. But if you ever have been on the ship, try to tow a net day and night, it's not only physically challenging, but I can tell you how accurate it is. So we need a really sense of revolutionary the sampling method. So this is what we really, really need your help, because without your help, and whatever the models we built, whatever the prediction we made for the climate change, and then we cannot really tell you how much we can trust those estimates. So we really need your help. Okay, so, and don't forget, when you build a sensor, we have to think about this, all possible platforms. And we think in last many years, and we talk about putting into cabled moorings or the cable observatories. We talk about the automated and Lagrange platforms like drifter, floats, glider, AUV, and ROVs. And we talk about put on the fixed stations like moorings and oil platforms. But we do not forget, we have many, many container ships that are going around in the ocean. And we can put all those sensors onto those ferries, containers, which can really also help us take a lot of measurements. So I really encourage engineers and technicians to really think about this, how to advance our sensor system and not only measure our state variables, also think about this process rates. And not only think about this to put onto rosette, put on the container ship, but also think about this, put them onto gliders, AUV, those small sensors. So we really have challenges in engineering. So as a final remark, they are urging the need for new sensors and integrated sensors to monitor environmental state variables so that we can know the world is changing. And then we need to resolve rates and the changes and the rates of the process. So without we know these rates, we won't be able to really validate our models. Because all the models, we're always making jokes about it. You have like 10 state variables, there are 100 rates, try to fix these 10 variables to predict. But none of the 100 rates are measurable. So that will become really challenging. So this is why we really encourage engineers to help us to design sensors we can measure rates. So we need to take a holistic approach to resolve integrated environmental processes, because only measure one variable, one process is not enough, because they are all coupled. We need to take a holistic approach of the sensor system. So in the end, I say a good instrument will strengthen our capability to understand and predict changes in environment, climate, and resources. Thank you very much.